The headline of our news today is all about the citric acid cycle, or which is also known as the KREBS cycle. And for our daily news, we have the event in the electron transport chain as well as the ATP synthesis. But before we proceed with our headlines, we need to go back to the incident in which is the preparation for the citric acid cycle. So going back to the previous topic, we begin with the glucose, a molecule which is composed of 6 carbon chain, and through a process of glycolysis, which was reported by the recent reporter, it was split into half, which is called the pyruvate. Obviously, it is composed of 3 carbon chains or a 3 carbon molecule, as the glucose was split into half. So a glucose is a 6 carbon chain, and when it is split into half, it becomes 3. So the pyruvate gets oxidized, and it is called the pyruvate oxidation. It cleaves off one of the carbon, and so now we will have 2 carbons left, which will be the acetyl-CoA. Going back, the citric acid cycle, which is also called as a KREBS cycle, was named after the one who discovered it, biochemist Hans Krebs. So this cycle happens in the innermost part of the mitochondria, which is the matrix. Everything that happens in this cycle is an enzyme catalyzed reaction. So there are enzymes that we will encounter later on. So there are 8 enzymes that are utilized in the KREBS cycle, and these are the following. You can see it in the presentation. So what we had discovered earlier was just the preparation stage of the KREBS cycle. We ended with the production of the acetyl-CoA, right? So the acetyl-CoA is the fuel for the citric acid cycle. So first, let's have an overview about the cycle. We begin with the acetyl-CoA merging with the oxaloacetic acid. It produces a citric acid, and it will be catalyzed by different enzymes going through a bunch of steps, which makes it into different kind of intermediates, and then it goes back to being oxaloacetic acid. So now it makes sense why is it called a cycle, right? From the merging of acetyl-CoA to oxaloacetic acid into becoming a citric acid, and then going back to oxaloacetic acid. So let us now discuss what are those bunch of steps in the citric acid cycle. So the first step will be the acetyl-CoA becoming a citrate. So the enzyme citrate synthase catalyzes the condensation of acetyl-CoA with oxaloacetate, which produces the molecule citrate, or also known as the citric acid. In this step, it requires 1 water molecule. Note that the citrate is a tertiary alcohol, that is why it cannot be oxidized very easily. In the second step is the citrate to isocitrate. So the enzyme aconitase catalyzes the dehydration of the citrate, producing an intermediate molecule cis-aconitate. And then it is hydrated, which means that a water is required. We can see here that here is the enzyme aconitase and a sign of water, as it requires water. So in the third step, we have the isocitrate to alpha-ketoglutamate. The enzyme isocitrate dehydrogenase catalyzes the dehydration of isocitrate, producing an oxalosuccinate, and then it undergoes into further process to become an alpha-ketoglutamate. And in this step, 1 NADH is produced, and as we can see in the illustration, the molecule NAD plus is reduced into NADH. And then a molecule of carbon dioxide is released, producing into proceeding into the electron transport chain. And for the step 4, we have the alpha-ketoglutamate to succinyl-CoA. It is catalyzed by the enzyme ketoglutamate dehydrogenase, decarboxylates alpha-ketoglutamate, producing 2-succinyl-CoA. In this step, 1 NADH is produced and another molecule of carbon dioxide is released, proceeding to the electron transport chain. And this reaction is catalyzed by the molecule alpha ketoglutarate dehydrogenase. In the fifth step, we have the succinyl-CoA desuccinate. It is catalyzed by succinyl-CoA synthetase, and as we can see that a GDP is reduced to GTP, and so it becomes into an ATP and reduced into ATP. In the sixth step, we have the succinate to fumarate. The succinate dehydrogenase oxidizes the succinate molecule, producing a fumarate. In this step, the FAD molecule is reduced into FADH2 as the enzyme eliminated the 2 hydrogen. And step 7, we have the fumarate to malate. It is catalyzed by the fumarase and then it becomes into malate. And in this step, 1 water molecule is required. We can see here the arrow of the water as it requires water molecule. So in the last step, we have the malate to oxaloacetate. So the molecule oxaloacetate regenerated from malate, which allows the cycle to start anew. It is catalyzed by malate dehydrogenase and produces 1-NADH from the reduction of NAD+. So to review the cycle, we can see here that it undergoes into a lot of process. Starting from the acetyl-CoA, which is a 2-carbon chain, and oxaloacetate, which is a 4-carbon chain, it reduced into citrate, which is a 6-carbon chain. We can see here in the process that in the isocitrate to alpha ketoglutarate, it released 1 carbon chain, which is the carbon dioxide, and into the alpha ketoglutarate into succinyl-CoA, it released again 1 carbon chain, which is a carbon dioxide. And then we can say that there are 2 carbons that are cleaved off, and so it goes back to the oxaloacetate, which is a 4-carbon chain. So as the citrate is a 6-carbon chain and then cleaved off of 2-carbon chain, and so it becomes 4-carbon chain, the oxaloacetate. So the products of this cycle are 1 ATP, 3 NADH, and 1 FADH. So the KREBS cycle occurs in every pyruvate. So there are 2 pyruvates, right, in the glycolysis. So let us multiply it into 2. That is what is equal to the products. So there are 2 ATPs, 6 NADH, and 2 FADH. So these products of cycle leads to the production of ATP. The molecules that are transported to the electron transport chain is used in order to produce ATP and other molecules. This cycle is the main source of energy for cells and the important part of aerobic respiration. The Krebs cycle is important because it releases energy that cells need for cellular activities and reactions. The energy released can be used to produce ATP through chemiosmotic coupling for cell processes, such as protein synthesis and motor activities. For example, the acetyl-CoA is made by the Krebs cycle and sent to the electron transport chain, where it releases energy for chemiosmotic coupling, leading to ATP synthesis. The enzymes control every chemical reaction that takes place in biological organisms, and this particular chemical organize and deorganize molecule into more complex molecule, allowing for different responses to occur depending on what they're doing at the time. So the Krebs cycle enzymes are carefully organized as many reactions as possible can take place at one time, minimizing the number of steps required and increasing its efficiency. So that would be all for the Krebs cycle. Now let's proceed to the next reporter, which is Crystal Sumagpao.
And for our second news, the electron transport chain. The electron transport chain is a series of four protein complexes that couple redox reactions, creating an electrochemical gradient that leads to the creation of ATP in a complete system named oxidative phosphorylation. So at the heart of this system are four protein complexes numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes number 3 and 4. Proton pumping requires energy, and the four protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the four complexes are collectively referred to as electron transport chain. So as you can see in our diagram here, let's focus on complex number 1. This is a byproduct of sugar metabolism called NADH or NADH deposits, which two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why. So an electron moves from the top redox center to the bottom. So first, the bottom redox center has a higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. So a small amount of energy is released each time an electron is passed between redox centers, and complex 1 harnesses this energy across all the redox centers and uses it to pump protons. And the last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. So in complex number 2, this is similar to complex 1 in two important ways. First, high energy electrons also enter complex 2 via byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. And coenzyme Q molecules from complex 1 and complex 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through two redox centers before reaching cytochrome C. And cytochrome C carries the electron to complex 4. And the electron transport chain ends in complex 4, where a series of reactions involving 4 electrons convert a molecule of oxygen to 2 molecules of water. Crystal Sumagpao reporting.
ATP, the electron transport chain, serves to set up a proton gradient. This gradient exists between the intermembrane space and the inside of the mitochondria, or the mitochondrial matrix. We can characterize this gradient in the following way. A high proton concentration exists in the intermembrane space. A low proton concentration exists in the mitochondrial matrix. Protons are pumped from inside across the inner mitochondrial membrane by the protein complexes 1, 3, and 4 in the electron transport chain. This establishes an electrochemical gradient across the inner mitochondrial membrane. There is but one purpose for establishing this proton gradient. It allows the free energy liberated in the electron transport chain to be stored in electrochemical gradient called the proton motive force, or PMF. This analogies to the storage of electrical energy in a battery, which is done using an electromotive force. Note that a region with high concentration is a region with high electrical potential. A region with a low concentration is a region with a low electrical potential. The return of protons to the mitochondrial matrix, which is accomplished by proton translocating ATP synthase or complex B, provides the energy that directly drives ATP synthesis. ATP synthase has globular units called F1 complexes that protrude from the matrix side of the inner mitochondrial membrane. The F1 complexes are also called ATPase, and they have subunits that can hydrolyze ATP but not synthesize it. The F1 complex is bound to membrane channels called the F0 complex. The F0 complex function as a proton pump in which bring protons back from the enter membrane to the mitochondrial matrix. The F0 works in concert with the F1 subunits to produce ATP. Three B subunits appear to work together. One binds ADP, one binds ATP, while a third is empty. This is not static situation, rather each subunit cycles through different states with the transformation between each state made possible via the energy obtained from proton transfer. Hence, conformational changes in ATPase driven by proton transport appear to drive the synthesis of ATP. Protons are transported through the F0 complex, causing conformational changes in the three B subunits. Three states of a B subunit appears possible. These are denoted to the tight P state, the loose L state, and the open O state. The binding chains mechanism as seen from the slide of the F1 complex. There are three catalytic sites in three different conformations: loose, open, and tight. For clarity, only the three V subunits are shown. Substrate ADP plus PI initially binds to the open site and is converted to ATP at the tight site. In step 1, rotation of the Y subunit causes a conformational change, resulting in a change in the formation of the sites. As a result, ATP is released from the enzyme. In step 2, substrate again binds to the open site, and another ATP is synthesized at the tight site. This is Aliyah Mapandi reporting.